All right, good morning, everyone. Welcome to our Tuesday Masonic Cancer Center seminar series. We're very fortunate to have with us an exciting speaker today. My name is Chris LaRocca. I'm one of the surgical oncologists here and oncolytic virotherapy researchers. Our speaker today, Dr. John Bell, received his Ph.D. from McMaster University. He completed postdoctoral fellowships at the University of Ottawa, as well as the National Institute for Medical Research in London. His first faculty position was at McGill University, and he later moved on to the University of Ottawa, where he currently is, and serves as a professor in the Department of Medicine, in addition to a senior scientist at the Ottawa Hospital Research Institute. He's received many accolades and awards over the course of his career, including the Queen's Diamond Jubilee Award, Fellowship in the Royal Society of Canada, and most recently, the Dr. Chu Wai Memorial Prize in Cancer Research. He's had extensive history of funding as well as high impact publications and trained a number of graduate students as well as postdoctoral researchers who have they themselves gone on to make contributions to the field of oncolytic biotherapy. And so it's my pleasure to invite Dr. John Bell up to the podium to give his lecture in synthetic virology strategies to create cancer therapeutics. So please help me in welcoming Dr. Bell.
Well, thanks very much, Chris, for inviting me and giving me the chance to visit here. I have heard about this place, but never actually been here, and it's sort of cool to see. I'm looking forward to the day actually to visit with other people as well. Um, and I was really pleased that you arranged for it to be freezing cold, so I could actually start to break into the winter mode. So this is actually downtown Ottawa. And just to show you where we are over here. Let me see if I have the laser on. Yeah. Over here is the Parliament building. So that's where the government sits. And we have this really nice canal that runs down the middle of the city, which is about 6 kilometers long. And you can see all my postdocs have boats here that they get to drive to work in. And and people actually go to work in boats every morning or kayaks, which is sort of cool. And this is of course what it looks like in the middle of January, but actually not really. In the middle of January, it's more like this, and it is quite cold in Ottawa. But what's nice about it is this canal freezes, and people skate up and down it. You can see there's lots of little places to go and buy. We call beaver tails, which are sort of deep fried donuts, sort of covered. They're quite delicious, especially when it's minus 30 out. But people skate to work in the morning too, which is pretty cool. Anyhow, if you had a chance to come to Ottawa, you should drop by. It's a pretty cool city. Things I'm going to talk about today, I just want to mention that some of these discoveries have been incorporated into a company called Turnstone Biologics, which is developing some of the things I'll talk about today. So the first thing I want to do is I want all of you to really appreciate this PowerPoint, because I spent about an hour trying to make this slide, and it's incredible animation. So prepare yourself for this. So, the challenge of systemic therapy for cancer is that if we use something like chemotherapy and there's a tumor here, what you have to do in order to get that appropriate dose of the tumor, was that impressive or what? I mean, that's so is to really soak the person in the chemotherapy. And of course, that's one of the problems we have, is that they have drug everywhere in their body at high levels, and it leads to toxicity. So the real dream that we've had for many years is to be able to have replicating therapeutics. So instead of having the therapeutic have to reach high concentrations throughout the body, the idea is that it actually will find the tumor, grow within the tumor, and produce the therapeutic dose there. So that would really be, I think, the dream we've had for many years. And of course, there's lots of ways to do this now. Through immune cell therapy is another example of a living drug. Uh, Various kinds of bacterial approaches are being used. And I'm going to talk today about the application of viruses for this purpose. So I'm going to talk about viruses today as replicating cancer therapeutics. So just to sort of get everybody on the same page of how we view this and think about it, the last 30 years of molecular research has taught us that a lot about how cancers evolve, and they go from normal healthy cells to frankly malignant tumors. And along that pathway, there's a lot of mutations or epigenetic modifications or whatever that happen that change this normal cell. And it goes things like disruption. We now know things like disruption of apoptotic pathways, metabolic deregulation is a well-known hallmark of cancer. Probably one of the most important ones is immune escape, the ability to cloak yourself and hide away from the immune system, so you can become this frankly malignant tumor. So we now understand a lot of these things. They don't necessarily happen in this order, but We know that these are the hallmarks of cancer, the things that have to happen for a normal cell to become a cancer cell. And I think, what became clear to us about going almost 25 years ago now is that malignant cell evolution and cellular antiviral responses are incompatible. So if you're a healthy cell and you get infected by a virus, you do all the things you can to protect the body because you're an altruistic cell. And so you'll stop growing, you'll perhaps commit suicide, you'll call the immune system over to kill you to stop the spread of the virus, and you'll actually block angiogenesis. So you do all those things so that the virus is limited and not able to spread very well. And if you're a cancer, that's just the opposite of what you want to do. So cancer cells never want to stop growing. So whether they're infected or not, they don't want to stop growing. They don't want to commit suicide ever. They want to they're selfish. They want to continue to grow, and they want to hide from the immune system. So all these things that normal tissues use to fight virus infections, cancer cells don't want to do those things because they basically have become immortal, and want to avoid the sort of pathways that are often used to fight virus infections at the cellular level. And so I would just argue that we've recognized over the last, I said, 25 years or so that cancer cells are genetically predisposed to virus infection. So you can hypothetically, in principle, you can use a virus to preferentially attack and kill cancer cells because they are genetically more sensitive to virus infection. And this sort of led to this concept of an oncolytic virus paradigm, where a virus, when it enters a normal cell, will trigger all the events the normal cell has in place, all the sensors to say, oh, I've been infected, and it'll turn on an antiviral program which will stop the spread of that virus. On the other hand, because cancer cells have turned off all those sensing and signaling pathways, the virus will be able to spread and go to other cancer cells and destroy them. So this is really I would say is the primary selective way that oncolytic viruses are developed to select between cancer cells and normal cells, and that is we create viruses which are able to infect cancer cells and take advantage of the fact that they don't have antiviral programs, but when they enter normal cells, they're attenuated and are stopped by the antiviral programs in the normal cell. So that's really what we were thinking about, like I said, almost 25 years ago. But what we recognized too is that although I guess the original dream was we can make these replicating therapeutics, they'll go and they'll rip through the tumor and destroy it and get rid of it, we'll all be happy. But we recognized as time went on and we did more clinical studies that was not going to be the case, that viruses are unable to actually completely eradicate tumors all alone in most people. But what we did also discover as a field is that, in fact, they're much more than cytolytic agents. Because of the fact that they are able to set up an infectious front at the cancer site, they also potentially can alert the immune system to come in and recognize that the cancer is not supposed to be there, begin to recognize various kinds of cancer antigens and destroy them. So it became obvious to us that viruses could be replicating cytolytic agents, but also more than that, they're immune stimulating agents and potentially could be used or engineered to really potentiate that activity. So we tested a number of viruses in the clinic over the years. One that we did early on was a Vaccinia virus called Pexa-Vec, as shown on this slide. This is a gentleman who received Pexa-Vec. He had widespread disease. He actually had liver cancer. And when his tumor was injected with this Vaccinia-based virus, he had a great response. But this was really what I would call an elite responder to this infection. Most patients did not have this kind of response. And so we recognized that although there was potential for viruses to be effective therapeutics, there was work to be done. There's things we had to do to make them even more effective. And our dream was to actually get more patients to respond like this gentleman who had a really widespread complete response to his disease, even though he was an advanced stage patient. So why is it that not more patients respond like this? What was the challenge? And I think it also became clear from lots of different work done around the world that it's really tumor heterogeneity which prevents us from having a single monotherapeutic strategy for treating cancer. We're not going to find, in my mind, the magic bullet that all on its own is able to fight cancer in every patient because of this heterogeneity. And this is just an example of immune heterogeneity in cancer cells and different tumors. So there are some tumors which have what we call an inflamed phenotype. They have pre-existing immune cells within the tumor. They can be perhaps activated by immune checkpoint inhibitors and have good responses. There's other kinds of cancers in which the immune cells can get to the tumor but get blocked at the outside, and these are sort of excluded tumors, they're called. And finally, there's others that are really immunologically ignorant, immune deserts, as Ira Melman has said. And so you can see that these are approximate proportions I just sort of made up, but I think it's probably true that about 20% of cancers are in this sort of phenotype and the rest are in these two. And so you have to do other things to try to get those cancers to respond to the kind of therapies you're developing. And over the years, many people have tried lots of different things, and we're all familiar with this, using various combinations of therapies to try to push tumors like this over to this state where they can respond to things like immune checkpoint inhibitors. And what that really says is that we really need to have combination strategies of some sort in order to deal with this incredible heterogeneity that we see within and between patients. And this is just one example from Sam Rabkin's group that was published a few years back, just sort of bring home that message. He was creating an oncolytic herpes virus that expressed IL-12. And, the dream was we'll have this virus go in, express IL-12, and everything will be great. And of course, that's not what happened. So then he started to try to overlay things on top of that. And ultimately, you can get to a situation where you can have a complete response in almost all of the animals that were treated in this particular model, but it required overlaying anti-PD-1, anti-CTLA-4, IL-12, and oncolytic virus infection to make that happen. So I think this just illustrates that, again, because of this heterogeneity, we can't go in with a single approach and not and hope to have good responses in most patients. So what that says, but the one thing that tells you is that if we have to start to overlay multiple therapeutics on this, it's going to really be overwhelming to our healthcare systems, because some of these great new therapeutics cost hundreds of thousands of dollars a year per patient. And if you have the frequency of cancer that we have in North America and around the world, that's just going to be impossible for us to sustain if we have to actually treat people with multiple therapeutics. So what can we do to try to deal with that problem? There's lots of ways one can imagine getting around that, but the thought we had was, can we make basically one therapeutic that has multiple weapons in it to attack cancer? So can we build an immunotherapeutic battleship, basically? And what this would be is a vehicle, in the case of a virus, which is very potent, is very targeted, is weaponized with multiple therapeutic transgenes. And the platform we decided to work on was Vaccinia virus. We'd had a lot of experience with it in the past, and we thought it had a lot of the virtues that would make for a good platform for building out multiple therapeutics within one therapeutic. And the dream is you could create a virus like this, which is multiply armed, and then you really only have one therapeutic, but it has multiple activities that you can control and regulate. So I like Vaccinia for a lot of reasons. I mean, there's lots of great oncolytic viruses out there, and lots of them have really interesting properties. I'm going to argue that Vaccinia is a good choice for today's talk because it is very safe. So Vaccinia virus, as you probably all know, was the smallpox vaccine. So it's been in the human population for hundreds of years, and we know a lot about how it behaves in the human population. It has very broad tumor tropism. It really can infect just about any kind of cell you can think of. And so because it can infect any kind of cancer, it's really a matter of tailoring it to be selective for the cancer. As I said, exploit the defects that are found within cancer cells to do that. We know that it can be given as a systemic therapeutic, which I think is an important aspect for trying to treat patients with metastatic disease. And this is just an example of, this is again, I mentioned Pexa-Vec, which was a virus we developed many years ago now, and is still being clinically developed by another company. And this is an example of two patients that were treated in Ottawa. This was a colon cancer patient and an ovarian cancer patient. And we gave the virus intravenously, and then we did biopsies of them about 10 to 10 days later and stained for virus antigens. And what you can see in these two samples is that we see where there's brown staining is actually in the various malignant tumor glands, and in the intervening normal tissue, it's blue staining, and there's no virus replication. So this is giving the virus systemically. I think this is around 1E9 PFU per patient. And you can see a week later that it's replicating nicely within the tumor bed itself after systemic delivery. So in principle, this is an agent you can give intravenously, and it can, in principle, find all the tumors within the body and replicate and hopefully be therapeutically effective. So that was one of the things we really liked about Vaccinia. We can still continue to try to develop. Vaccinia is a really interesting virus. I'm not sure how many of you know much about it, but it's almost a cell. It's so genetically complex. It's got lots of things it does. It has a really interesting life cycle. It enters into the cell, It goes through a lot of different aspects of its replication. It even makes its own membrane factories. It has multiple isoforms it makes. It makes one isoform that does what's called surfing, where it goes from one cell to the next cell. If that cell's already infected, it surfs over it to the following one, so that's pretty clever. It has another version of itself, which has a second membrane on it to help it hide from the immune system. So it does lots of clever things. It's a really, really interesting virus from a biological perspective. And I think also from a therapeutic perspective, if you can understand it well, you can then exploit some of its biology for therapeutic gain. It's a member of the pox virus family. So, pox viruses sound a bit dangerous. And in fact, smallpox is pretty bad. But there's lots of versions of this throughout nature. So it's a virus which has adapted itself well to lots of different kinds of infectious agents. The area I'm going to focus on today is just the Vaccinia virus aspect, which are here. And these are various kinds of viruses that have been used largely as again, smallpox vaccines. So you might recognize some of these names like Copenhagen Lister smallpox vaccines developed in Europe, and Wyeth is one that was used in the United States. That's just down here. And they're all genetically similar, but also have unique properties to them, which we've been able to exploit. So we've got these viruses. They're really good at infecting people and infecting animals, and they've got a lot of interesting biology. But what we want to do is be able to tailor them to our use. And so I would say you've got a virus which is really an interesting biological agent. It's more like this sports car here. It's got a lot of really sophisticated properties, abilities to attack people and animals in different ways. What we want to do is really just strip it down to its chassis, take out all the extra features that are not necessary, so we can actually build up from that chassis the kind of therapeutic we're trying to develop. And as a virus, it's quite complex. The genome can be over 200 kb. You can have 200 different genes involved in interacting with the host cell, so it's quite a complex virus. And so really, this is the sports car model. What we want to do is figure out what don't we need for this to be an oncolytic virus, and what can we use to make it, and what can we add to it to make it more effective therapeutic. And so we really need to have a better understanding of the genetic elements to control the ability of this virus to infect and regulate its activity within cells. So we wanted to interrogate the genome. And our thought was, can we use a way to sort of randomly mutagenize this virus and find genes which are necessary and genes which are not necessary for it to be an oncolytic agent? And so the strategy that one of my students took was to take and use retroviral insertion, or transposon insertion, I should say, to try to mutagenize the virus by just randomly integrating it. And the idea was let the transposon enter. It goes into TTA sites. If it turns out that you insert into a critical gene, then that virus won't be viable and you won't get a plaque. If you've inserted in a gene which is not necessary, then that virus will remain viable. You'll collect it and you'll be able to see where that insertion site is. So it's a way to actually interrogate the genome. We've done this now with Vaccinia virus and also with herpes virus, which is even more interesting. And we're beginning to get some really interesting ideas about what's the minimum chassis we need to make this an interesting therapeutic. So this analysis was done, and just on this slide here, it's just a heat map. And let me explain to you what it is. These are different genes in the top in the Vaccinia virus genome into which the transposon could insert. And down here is different cell lines that we tested the library of insertion mutants on. And if you see red, that means that this gene, if you insert it into it, would kill the virus. It wouldn't be able to replicate in any of these cell lines. If you see green, that means you could insert into this gene or the gene beside it here, and that virus grows perfectly well. It's not impacted in any of the cell lines we looked at. So by doing this kind of screening, we're able to really elucidate where in the genome we had genes which were essential for replication in cancer cells, and ones which are dispensable for that, and ones which maybe give you the selectivity you want in normal cells. So just to give you an idea of how this played out, this gray here band is the potential insertion sites of a transposon in the Vaccinia virus genome. So any place there's a TTA sequence. So in principle, you could insert into every single one of these sites. You could at least do that biologically, but could you rescue a virus? And what we looked at was, where did we actually insert in our rescued library? And what we found was really, I think, quite interesting, where The red dots represent the frequency with which we saw an insertion event. And you'll see that if you look along the genome, although you have lots of potential insertion sites here, very few red dots. The red dots tend to be clustered at the two ends here. And these two ends are places where what it tells us is that we could insert into that part of the genome and the virus remain viable for infection in cancer cells. Whereas if we inserted somewhere in here, those viruses were no longer viable. And so it really told us that we could geographically sort of figure out where there are genes which are necessary and where other genes are not necessary for creating an oncolytic virus. And if you look at conservation between pox viruses, this part of the genome is really highly conserved, not so much conserved down here. And what that tells us is these regions of the genome here are more involved with how the virus interacts with the cell. And these parts in the middle, this is the sort of chassis that we were looking for, the part of the virus which has to be maintained for it to be a replicating therapeutic. So we felt by doing this, we really identified the sequences which made let me just close this down, which made this a virus product. And we could now take that information and build upon it to make it a more effective therapeutic. Oops. For some reason this stopped. Oh, here we go, maybe. Okay. And by integrating that data, we came up with a new genome. This is we call the superior killing virus. It had a lot of different reasons that we called it SKV. Most of them I can't tell you what they were, but these are things my students made up. But I ended up saying, no, we're going to call it superior killing virus. And this genome is very similar to the previous one I showed you, except now it's had about 25 kilobases removed from it. And this is cool because it meant we now had a virus product that had an extra 25 kb of space to stick in new genes that we might want to have that are therapeutically useful. So just to give you an example of how this virus is a better oncolytic in our opinion, is if we looked at this again is Pexa-Vec, the one I mentioned to you at the very beginning that we used in the early studies. And these are a number of patients that were treated with Pexa-Vec. And you can see occasionally they would get these lesions on their skin. So it was still pretty safe, and these lesions would be resolved, but they were clearly pox lesions that formed on these patients, as well as the fact that we saw infection of the tumors. And what that means, if you're trying to create a more potent virus, one that's going to express something really interesting like IL-12, you really don't want to infect any normal sites, because even if it's only a rarely infects a normal site, it's going to be a problem. You're going to create an issue for the patient. So this kind of off-target effect was really something we didn't think was acceptable, even though the patients did quite well otherwise, because we felt it was a situation where one, in fact, might actually have off-target effects that you didn't want to have. So we've now gone into people with SKV, but just these early studies, we had a sort of a surrogate for this kind of effect. If we looked at, these are tails of mice here, and I think you can see there's little red lumps along the side. They have pox lesions on their tails. So this is using the Pexa-Vec virus we started with originally. This is the parental strain of SKV, but you can see SKV, it's got nice smooth tails. They're very little, just like PBS treated animals. There are no pox lesions. So this has become extremely selective by the deletion of the genes that we've taken out of it. If you look at, again, after systemic delivery, if we look at Pexa-Vec or other kinds of Vaccinia viruses that express luciferase, you see that after intravenous injection, these are nude animals, you see a lot of off-target infections of tails and snouts and so on. But with SKV, it's really restricted only to the tumor bed. And these signals seem a little bit lower than here, but in fact, that's because these tumors are resolving away. So we have a virus which is extremely selective. Even in nude animals with no immune system, it really only infects the tumor and not the normal tissues. So we like this virus platform now because it's something upon which we can build. It's highly targeted for replication within the tumor and not in normal tissues, which is one of our goals. It has an inherent ability to stimulate anti-tumor immune responses, which I'll talk about a bit more in a second. And that's because we've deleted genes, which the virus are virulence genes, which the virus normally uses to antagonize the immune system. It's been engineered for better spread between and within the tumor itself, which I'll speak a bit about. And we've shown it can be delivered systemically or by direct injection, of course, and it has broad tropism for many kinds of human cancer cells. And because of the deletions we've made, we now know we have about 75 kb of cloning space. So we can really add large modules to this virus if we want. So the real dream is to create a virus which can do things like express chemokines that could attract T cells, for instance, to the tumor. Maybe express, this is a term that was developed by Cioxis. These are membrane-bound T cell engagers. So maybe get a mite to get the T cell to recognize the cancer cell as foreign. Express bites, for instance, T cell engagers to bring in T cells to tumor cells. Perhaps express immune checkpoint inhibitor antibodies. So you don't necessarily have to add those systemically. You can actually just express them within the tumor bed. So that's sort of the dream of where we're going with this. So we're really trying to rethink of how we use oncolytic viruses and not think about them as only cytolytic agents, but actually more like things that one virus can create multiple therapeutic payloads. This idea of immunotherapeutic battleship, or a mobile pharmacy that goes around and dispenses the drugs where you want them, when you want them. So the first agent that we've developed that's gone into the clinic is one in which we express a FLT3 ligand, which is used to activate dendritic cells, IL-12, which is a well-known potent stimulator of T cell activity and other immune things we want to see happen within the tumor microenvironment, and ipilimumab, or anti-CTLA-4. So we were able to include the entire antibody within the backbone of the virus. And the reason we did this is we wanted to sort of attack many aspects of the immune cycle, as has been coined by Ira Mellman. And so this virus has now gone into clinical studies, and it's still an ongoing phase 1 study. But I can tell you we've seen some really interesting things where the virus has gone where it's supposed to go. It's recruited T cells into the tumor microenvironment as it's supposed to. It's been really safe, even at higher doses than we use with Pexa-Vec, which is great. And we've had some patient responses, including one recent CR. So we're pretty excited about this, although it's got a long ways to go. That's sort of where we're going with this kind of product. We're thinking we're going to make something, as I said, it's going to be like a mobile pharmacy where it goes and delivers things like anti-CTLA-4 only where you need it, not systemically, but only within the tumor. So that's an ongoing part of the project I'm not going to talk any more about, but I can certainly answer questions if you'd like later. What else do we need to do to make this a better kind of therapeutic? And certainly extracellular matrix is a huge problem for in terms of cancer treatment. It's a block to chemotherapy, it's a block to immunotherapy, and it's really a major characteristic of almost all tumors. And it actually makes up a huge mass of the tumor itself. And so you sort of think about it, it's like this: you're trying to get through this forest, and there's all these trees. So what you really need is some sort of a chainsaw to cut your way. I'm sure in Minnesota that you all have your own chainsaws at home. So, this is exactly what you want to do: is you want to cut through that forest and get your virus to where you want it to go. So we've looked at a number of different ways to do this, and it's been work done by other groups as well. Ramon Alemán in Spain has developed an adenovirus, which is now in a phase 1 study, which encodes a heparinase and that looks really sort of provocative in early studies. So we've looked at a bunch of different things. I'm just going to talk about 2 today. One is this collagen degrading enzyme called CoLH. Another one is a degrader of hyaluronic acid, SPAM1. What's interesting about this, both these enzymes are already approved by the FDA to give as therapeutics as enzymes themselves. So we think there's a good track record there for encoding them inside of viruses. And so here's just a couple of examples of how these can work. In this case, the virus itself expresses GFP. So when it replicates productively, you start to see more GFP. And this particular kind of spheroid, you can see that if we just add the base virus itself, it really has a hard time initiating infection in the spheroid. There's a little bit of green, but you can't really see much here. In this particular cancer, if you have a CoLH enzyme, you see that in some of the samples, you begin to see some green, but the heparinase is really effective and allows us to get widespread diffusion throughout the tumor. So our thought was, can we put more than one enzyme into the backbone? We have 75 kb of cloning space. And so even though you have heterogeneity between patients, you'll have, if you have a lot of collagen, you'll have CoLH. If you have a lot of hyaluronic acid, you'll have heparinase. That's sort of how we're thinking about this. These are just tumor explants. These are directly from patients. So we have a great setup with a colleague of mine, a surgeon, Rebecca Auer, who has set up a really nice system for allowing us access to patient samples on basically a daily basis. And we can look at a variety of different kinds of cancers. And what we do is basically get a surgical specimen, and we have ways of processing that. And we can infect it, right away and look over the next couple of days how well the virus replicates or spreads or recruits T cells or whatever we're looking for. And so here's an example of a ovarian tumor core, which the parent virus doesn't really impact it very well. But if we use the virus which has COL-H or SPAM1 in it, we see actually we got good spread of the virus throughout that. Here's another one. Here's a lung tumor core. Again, SKV is not very effective, but we add in COL-H. It's a little bit better, but SPAM1 is really good. Here's an endometrial cancer in which COL-H really helps a lot, but the SPAM1's not as effective. So this sort of makes us think that it makes sense to have a virus which has multiple enzyme degraders in it, so it deals with that heterogeneity we see in patients. And what was, I think, provocative, and we still have a lot of work to figure this out, is if we take in this case, this is an A549 xenograft. This is not a patient sample, but a cell line in a nude mouse or skid mouse. And we treat with one of these viruses, in this case, the COL-H virus, the collagenase degrading expressing virus. You can see that where we stain for anti-Vaccinia, we see lots of Vaccinia spread is also where there's very little collagen. And we believe that's because the enzyme is being expressed and chewing through this collagen. So it's sort of like a snowplow going through and allowing it to spread better. So that's still ongoing work, but I'm really excited about the possibility that we could really potentiate a lot of the things we want to do with these viruses by putting in a spectrum of extracellular matrix degrading enzymes to help them spread. Another challenge we all face in cancer immunotherapy is that, in fact, lots of times these tumors escape immune recognition by downregulation of MHC, for instance, or other parts of that pathway. And so a way to overcome that, of course, is to use BiTEs or T-cell engagers, which has been done by a lot of groups around the world quite effectively. Basically, a BiTE is made with the variable parts of an antibody, and you can create different versions of this. But the dream is that you actually bring a T-cell by binding to CD3 to a tumor cell, and this engagement leads to an immunological synapse, leads to killing of the tumor cell. And T-cell engagers are worked well in the clinic in some respects, but they always have the problem of they rapidly turn over. You have to give them systemically. You often have off-target effects. So again, this seems like something that a virus should be good at delivering in the local tumor microenvironment, perhaps get you the dose you need where you need it and not elsewhere. And so we've created a number of different kinds of vaccines encoding these BiTEs. And the dream, again, is that the virus will infect. Not only will it kill the cell it infects, but it'll also produce and secrete the BiTE that will lead to more T-cell engagement of uninfected tumors and give you a more effective therapeutic. So this is just an example in a colon cancer model of a Vaccinia virus expressing a BiTE against CEA. And the bottom line is, in this virus in which we encode the BiTE, we see tremendous activity. It works really well. So even with somewhat limited infection, because you're secreting this BiTE in the local tumor microenvironment, you're able to see a really good therapeutic outcome. What's nice, I think, about it, which I'm excited about, is we see a phenomenon called epitope spreading that you're probably familiar with. And basically that is we bring in T cells and engage the tumor because of the BiTE. But once those T cells are in the tumor microenvironment, they become activated to recognize other epitopes. And so if you look over here, for instance, when we go and look at animals that have been challenged, that have been cured, for instance, with a virus expressing the BiTE, and then we go and challenge them again, either with a tumor cell expressing the original antigen, which in this case is CEA, or tumor cells which don't express CEA, what we see is that these animals can resist challenge to both those kinds of cells. So what that tells us is the virus going in expressing the BiTE helps to alert the immune system, to educate it to now recognize that not just the antigen that the BiTE's directed against, but other neoepitopes that are in there. And I think that's really exciting because it suggests that you'll be able to maintain an ongoing immune response against the tumor. And we can also use this virus systemically, as I mentioned before. It's easy to deliver systemically, and you can see here these lungs have no tumors in them, and that just shows you that the virus, when given intravenously and expressed in the BiTE, is very effective as a therapeutic. So we've now made a number of BiTEs against things like carbonic anhydrase, which is a really interesting marker within the tumor, but you can't really make, people have tried to make CARs against carbonic anhydrase, but there's too many off-target effects. But with the BiTE approach, which is transient and localized, we think that's going to be a good antigen. So we've made that. Other kinds of antigens that we've looked at in the tumor microenvironment, which I think are really interesting. So that's an ongoing series of projects. I really want to talk about something I'm really excited about, and this is work which I think is really clever, really innovative, that I've done with a colleague of mine, Carolina Ilko, who now has her own lab also in Ottawa. And the idea here is to, as you all know, our cells produce exosomes, and exosomes are involved in talking to other cells within the body. Cancer cells produce exosomes. Those exosomes actually go and create niches, for instance, to allow the cancer to spread. There's lots of things we don't understand about exosomes, but it's clear they have an important role to play in cancer biology. They also play an important role in some diseases like chronic hepatitis caused by hepatitis C virus. And it turns out that the infected cells produce exosomes, which go and then create surrounding cells to be susceptible to virus infection. And that got us thinking, can we actually take advantage of that biology? Can we program our viruses to make things, to reprogram exosomes, so those exosomes go out and impact the tumor microenvironment? And we've got a lot of different ways of doing this now. I'm just going to give you one example, but I'm reasonably excited about this. So the idea is the virus comes in, for instance, it could express a microRNA, an artificial microRNA that we design in some way to attack a particular target. That microRNA gets packaged into exosomes. They can go to an adjacent cell and release that artificial microRNA, and that can impact the physiology of the cell. The virus can also spread and just by oncolysis kill adjacent cells. But what it's doing is creating a factory within that tumor to produce exosomes, which can then release and therapeutic microRNAs. So, for instance, there are genes out there we'd like to drug, which we can't drug, like RAS, for instance, are hard to develop drugs against. But if you can actually make microRNAs that can target a mutant RAS, then you have a way to therapeutically impact the tumor microenvironment. So anyhow, this was a really interesting project that came out of a screen that Caroline and I were doing, and just give you an example of how this can work. So here's an example of, in this case, we're using not Vaccinia virus, but vesicular stomatitis virus. It's a rhabdovirus, another one that we've used a lot and developed in the clinic a little bit. And Steve Russell at the Mayo Clinic is developing a VSV-based platform now. And so in this case, we created an artificial microRNA that targeted PD-L1. And the idea was, could the virus actually go in, produce these artificial microRNAs and spread them throughout the tumor and eliminate PD-L1 expression, so you wouldn't need to add in an anti-PD-L1 or an anti-PD-1 antibody. This is work done by Matthew Krupi and Giuseppe Pugliese, who's an oncologist in Italy, but came work to my lab for a year or two. And basically, this is an example of how this could work. We put, for instance, B16 cells in the upper part of a Boyden chamber here, and then infect it with this virus. The virus itself cannot get through the filter here, but exosomes are able to get through and impact this lower layer. And then we look to see, have we impacted PD-L1 expression? And you can see that if we use VSV at the top with no microRNA in it, we still get PD-L1 expression. But if we add the microRNA against PD-L1, we see a loss of PD-L1 expression. So what that tells us is that the virus has infected the upper layer. It's produced the microRNA. It's been packaged into exosomes. Those exosomes are able to go in down to uninfected cells and change their profile, which is, I think, pretty cool. And what you end up seeing in the end is the black line, of course, is the one we want to look at. We use this virus along with an immune checkpoint inhibitor. We see really good outcomes. So it says that it's possible to manipulate the exosome profile of infected cells and change the tumor microenvironment in ways. You don't necessarily have to have a microRNA that impacts tumor cells. It could be impacting immune cells, for instance, in the tumor microenvironment. So we're really to continue on to exploit this particular biology. And we've done things like, for instance, encoding the virus targeting ligands that can be put on the surface of the exosome and take to a specific site and deliver a specific cargo. So I'm really excited about this particular pathway, and I think it's going to be really interesting. The last thing I wanted to speak about briefly is that if you want to have viruses which express lots of interesting kinds of transgenes, it'd be nice if you could control it. And so we've been a lot of effort into controlling genes expressed in Vaccinia virus using TED on, TED off, QMATE on, QMATE off, and also split T7 promoters. And it's gotten to the stage where we are becoming very proficient at controlling the expression of genes. So just as an example, this is a D13 and critical viral protein, which we can turn on and turn off. And you can see that if we infect these mice, the tumors are infected. If we don't, in this case, have the inducer present, you don't see any infection. But if you then add the inducer afterwards, you start to see the infection spread, and you can also turn it off. And I think this is going to be really valuable as we go move forward, because you want to be able to say, okay, we've had enough CTLA-4 in this tumor, let's just turn it off. Or we want the virus to spread a little more vigorously for a while, let's turn on virulence gene. So by doing this sort of synthetic virology approach, we can regulate how the virus replicates and spreads. I'm just going to skip over that. And one last thing I just want to mention briefly is, we've talked about an immunotherapeutic battleship. What about making it more, as Nicholas Martin, my lab, suggested, an aircraft carrier, where this initial big virus delivers something smaller, another kind of virus or self-amplifying RNA. And what he decided to check was, is it possible to encode an entire other virus within the Vaccinia? This has been done by other groups before, or a self-amplifying RNA. And the way he's done this is quite nice, is he's put in, for instance, a self-amplifying RNA based upon alpha virus genome. And we've now tested a number of different kinds of self-amplifying RNAs. They all work quite well. And then have, for instance, structural proteins separated from the genome itself. So you express the packaging proteins and the self-amplifying RNA in the same cell which has been infected by the parental virus, which is Vaccinia virus. And so this has turned out to be quite interesting. It's another way to allow you to get spread throughout the tumor microenvironment of something like a self-amplifying RNA. You might want to use it, for instance, to program T cells to express CARs within the local tumor microenvironment. And so just to give you an example of how this can work, the green represents the parental Vaccinia virus, and the red is encoded by the self-amplifying RNA. You can see over time, this is able to spread quite rapidly and quickly throughout the tumor, even faster than the oncolytic virus itself. So that's sort of an interesting, I think, concept for another way to generate and deliver genetic packages throughout the tumor microenvironment. So really just to summarize, we see now that there's ways that we can control infection. We're lucky with this particular virus, we can give it systemically. We can get a foothold within the tumor. We can use our systems for controlling transgene expression to allow, for instance, more robust virus replication and virus spread. We can turn those off and on. I didn't talk about it today, but we have other genes we turn on and off to allow the virus infected cells to escape immune recognition transiently, allowing the virus to spread and then turn them off again. So it's really, I think, becoming a different kind of world where we actually express multiple different kinds of transgenes and orchestrate their expression, the timing of their expression, where they're expressed, again, using drugs which are FDA-approved drugs. And so I think it's a way now to really orchestrate these therapeutics to be more than just cytolytic agents, but actually really important therapeutic delivery vehicles. And I'll just stop and recognize even a small number of the people who've been involved in this work over the years. And I've had lots of support from various kinds of funding agencies. So thanks very much for your attention, and I'd be happy to try to answer any questions you might have.
Thank you so much. It's a fantastic talk. So I wonder, what about the clearance of the virus, especially in human patient? Do you have that information? And do this deletion of all these 24 kilobase genes influence the clearance, the rate of the clearance of the virus?
Yeah, this is a really important question, because obviously the beauty of these therapeutics is that you can give them, as I mentioned, systemically. The downside of them is you generate immune responses against the backbone itself, and ultimately patients will develop immune responses and prevent multiple delivery. It's unclear, I'd say, in the clinic yet how that's clearly true in mouse models. We have treated patients who have been previously vaccinated against smallpox, for instance, and the virus is able to deliver quite easily. But of course, that vaccination was many years ago. But we have seen some patients in which we've treated them multiple times, and they still get delivery to the tumor. But is that a patient-specific thing? We don't know the answer to that yet. There's lots of people trying to create different ways to cloak the viruses, and that's obviously important. But what's really interesting is not just the delivery, but the persistence of the virus within the tumor can be quite variable between patients. We've had patients who we've treated, their tumors don't progress, but they don't get any smaller, and we biopsy them a year later, and they still have virus. So it's like it's created a little new ecosystem where the virus is there. Maybe it's hiding in the immune system, but it's not letting the tumor grow. So I think there's lots we don't know, to be honest, and this is an important issue that we need to work on for sure.
Thank you.
Mhm. Mm. Sure, yeah, happily. So these viruses don't selectively bind to cancer cells. They can infect any cell. So, but what's happened is, as the cancer evolves, say for instance, it gets rid of its anti-apoptotic genes because it wants to be a cancer. Apoptosis is a key way that cells clear virus infection. So a cancer cell has given up that ability. So as a result, it's like you threw seeds on the pavement on the grass. They'll grow in the grass. That's where the tumor cells are, but they won't grow in the pavement for the normal cells. Yeah, good. Thanks. Yeah. All
That was a really great approach, the insertional mutagenesis part, to try to understand what genes are critical for infection of cancer cells. But to me, it raised the opposite question: what do those genes do in the normal life cycle of the Vaccinia virus? Because they must do something.
right. Yeah,
And do they help you thinking about, generating the therapeutic? What genes could be further attenuated to make it less, reduce the toxicity?
right. Absolutely. So if they're in the virus has kept them for a purpose. There's no question. Viruses are very economical. So I think what they generally are genes that the virus needs to infect normal cells. And so if it wants to spread in a human, cause smallpox, whatever, it has a great complement of genes it uses for that. Those I would call virulence genes. Because the cancer cells are uniquely sensitive, we don't need those genes to allow the virus to infect the cancer cells. So by eliminating them, we allow the virus to grow in tumor cells, but not normal cells. But you're right, maybe they could be involved in making them grow in the tumor cells better. So that's where our thinking is around using controllable elements so we can turn them on and turn them off. So maybe we allow the infection to start, we turn on some of these genes, we let the virus start to spread, but if it starts to get off target a bit, we just take away the inducer and then the virus stays where it is. So it's absolutely true that those genes must play an important role in the pathology of that virus in nature. But in this system, we're creating an artificial system basically for, and the other thing is, a virus that you get an infection in nature usually is a pretty low dose infection, right? It's very little, but when you give it therapeutically, you're giving a large dose of virus to the patient. So by deleting some of those virulence genes, as long as the virus is located within the tumor, you're probably going to be okay.
Just while I'm asking, do you have an opposite research interest in trying to deliver genes to as gene transfer and other diseases outside of cancer? I mean, I think the tissue tropism in the skin is kind of interesting.
Yeah, absolutely. We definitely are doing those sorts of things too, which I think it's a really powerful technology for doing lots of things. So we have a lot of AAV things that we're doing as well. But I think the idea, for instance, I think of being able to deliver a self-amplifying RNA could be therapeutically useful in settings other than in cancer for sure. And so I think there's ways we can think about doing that.
Yeah, that was a really great talk. Thank you so much. I enjoyed very much. But having your experience, do you think it's really reasonable to control virus replication? The more I work with virus-based vectors, the more I understand that the main problem of oncolytic virus therapy is its efficacy, it's not safety. And do we want to sacrifice the oncolytic potential of our virus-based vectors? How important is that? Because as you mentioned, oncolytic virus does have the intrinsic selectivity, right? That prefers to replicate only in cancer cells. Any suggestions, any tips, any thoughts about that?
Yeah, you're absolutely right. I mean, I think the thing that has hurt the field, I think, is we've been so paranoid about safety that we've attenuated so many of our vectors that they don't work therapeutically anymore. So you're absolutely right. You can go too far in that direction and create a virus which lacks potency to do what it needs to do. So the sweet spot is to find it so it can differentiate between normal and tumor, but still has some oomph to it. And so that's why I think we have to sort of dissect on all these genes and figure out which ones to put back or perhaps which ones to regulate. And I like the concept of using regulatable promoters if possible, because you could then ramp the virus up and make it be more potent transiently, but then you can turn it off again. So I think, and Nino Kyoko, for instance, has done something like this with the nested promoter where he turns on ICP 34.5. So conceptually, I think there's a way to do this, that you can actually get the virus to be potent where you want it to be potent, but then tame it if you need to. Or maybe if you get lucky, the immune system will tame it for you. But that's, you've identified the biggest problem in the field is, and I think that's probably why there have not been more approvals of OVs yet, is because we have not gotten to the spot where it's potent enough and can deliver the things you want, but still remain safe. And so I think that's the challenge.
All right, any more questions? All right, let's say thank you one more time to Dr. Bell.